Hey there, for a video today we're going to be focused on scatterplots in Pluto. So in Pluto there are lots of different kinds of visualizations that you can create. I'm showing here a couple different kinds for RNA-seq experiments. We're really going to focus on scatterplots for this video and use them to explore how we customize different plots and different visualization parameters. So starting with this principal components plot here, which is a scatter plot showing different samples plotted on the X and Y axis with PC1 and PC2, I'll edit this plot to show off some of the different kinds of customizations that we can perform. So first of all, when I open up a plot, you can see here the plot itself is kind of in full screen mode, and I'm able to hover over the data points, which in this case are samples. I can always refer back to our sample data table here by scrolling down a little bit below, and you can see in this case that our PCA plot is showing 12 different samples. These were mouse samples taken at 3 different time points here, and then we had a time point here that indicated no stroke or no induced stroke. So one thing that I might want to edit initially on this plot might be the title. So I could call this PCA plot all mouse samples. We'll just give it a different kind of title here. And then it's also pretty common to show the proportion of variance measures on the X and Y axis. So I can check that box. And as I'm making any changes here to the plot, I can click Save Changes. Now we can see those changes that I just made reflected on the plot. So I can see the percentage of variance explained here on the X and Y axis and the new title that I had inputted. So for any plot in Pluto, this plot sidebar here is really where you'll find any kind of editable parameters that are really worth exploring for customizing the plot that we're looking at here. So I'm plotting PC1 and PC2, for example, but I could choose different principal components to show on this plot, manually set a start and end value for my X and Y axes if I wanted to. And then an important component of PCA plots in general is what kinds of variables are we grouping by or coloring by. So in this case, if I again refer back down to my sample data table, I'm referencing the time point column here, and that's how I'm coloring my different samples. So the 12-hour samples are colored in this darker blue color here, just based on the theme that I selected. I can choose to color by a different variable or even combine multiple variables. So if I want time point as well as gender, you can see that our little legend preview here has updated. All of the samples in this case came from male mice, so I'm not seeing any difference in the group numbers, but if I had female and male, I would see separate entries for 12-hour male and then 12-hour female. I'll just go ahead and keep these grouped by time point, and then you can see I can select from different default themes. So these will change the default colors, which again I'm seeing a preview of here down in the legend. I can select this one, for example, and then just click Save to see those values reflected on my plot over here. If I don't like the default theme colors or I do want to modify those further, I can always click on the color chips here. So maybe my NA value, or the no strokes in this case, I want to be black or gray. That's pretty common for the control group in your visualizations. And so I can click Save to see that again reflected on the plot. So now it's pretty clear here that my sham samples or my controls are down there. It's also pretty typical to show the control group listed first in your legend. So I can click on those drag handles here and reorder my legend items so that the NA or stroke or no stroke condition here is first. And then maybe I really want to clarify here that this is my control group. So I can rename that, give that a different display name, and click Apply, and then we'll save all of those changes. Great, so now we can see in our legend that we have control as the first group in our legend items here, and then we've got our color changes reflected, and we have the word control instead of the NA value that was being read directly from my sample data table here. The last change I'm going to make is just to order these groups by time point so that it's a little more chronological, 6, 12, 24, and I'll click Save. Great. So that kind of illustrated some different ways to customize a PCA, which is one type of scatter plot. To show some differences with another kind of scatter plot, we'll go over here to a volcano plot. A volcano plot shows the differentially expressed genes in this case, RNA-seq experiment. And instead of having principal components on our X and Y axis, we're looking at the log2 fold change value and the negative log10 adjusted p-value in this case. So we had performed this analysis in Pluto, and you can watch a separate video on how to do that. But what we're looking at here are genes that increased in expression, such as A2M, or decreased in expression in the particular comparison that we were doing in this group. So again, I'll start here by renaming the plot title so that instead of 24-hour versus NA, I'll call it our 24-hour stroke versus control. That just makes that a little bit more clear when I click update. I also like to show the line here indicating my p-value or level of significance. So I'll change that level of significance and then check this checkbox so that I can show that line on the plot. Now you'll see that that line has updated and my title has updated. You'll notice that the parameters here in this plot sidebar are slightly different than the ones that we were seeing for the PCA plot. So in this case, I can change the fold change, which are the x-axis values here, and even show those lines on the plot too. When I click update, now we can see vertical lines indicating where those fold change thresholds are. And I could make these more stringent. So just picking another arbitrary value here, you can see how that line shifted over here to the left. Again, I can manually set some axis ranges, select from different default values for the plot theme. Or for the volcano plot, I can manually set the color of a lot of different kinds of points here on the volcano plot. So for my significant up genes, which will be above this p-value line here and then to the right of the fold change value line that I've set, I can select a fill color and a border color. So I'll fill with a darker red and then have a border color of this lighter red here. And I can change point size and opacity and lots of other things too. For my significant down, same thing. I can select different fill color and border color, opacity. Maybe let's change the point size here just to show how that works, and then I'll click save. Now we can see that those points have been edited. Scrolling down a little bit more, you'll see that I can also label different targets on this plot. So I can manually search for different genes that I'm interested in. These are mouse genes that are available in this data set. Or I can highlight, this is pretty common too, to just highlight those genes anywhere on the plot here that we're seeing maybe some genes of interest. So in this case, pretty significantly upregulated genes in this particular comparison. Those were two different examples of scatter plots in Pluto and how we can edit different attributes on the plot using this plot sidebar menu here.